Welcome, everybody. We are all waiting for a cure for COVID 19 so we can all get back to our normal lives. Luckily, there are over 120 vaccines in development and testing, and over 200 treatments that are being explored to fight the COVID 19 pandemic. Today, we have a very special expert, Dr. Maria Elena Bottazzi, a virologist, microbiologist, and a distinguished professor at Baylor College of Medicine. She is also an expert in the field of vaccine development and has published several papers on coronaviruses. Dr. Maria, thank you so much for being with us today.
Thank you. It's a pleasure to be with you.
Thank you. So, you have worked with coronaviruses before, and you are aware of the new coronavirus. So, what are the differences between the previous coronaviruses and the new one?
Yes, that's correct. So our vaccine center, which we're based here in Houston, Texas, have been developing in partnership with a couple of organizations, um, um, trying to develop not only vaccines for SARS, also MERS, and of course now COVID-19. And as you imply, you're correct. The virus that is currently circulating and causing this devastation of a disease, it's actually very similar to the SARS virus that circulated back in 2003. We know that it has approximately 80% similarity within its genetic code. Both of the viruses use the same receptor to infect our human cells, which is the ACE2 receptor. And albeit it has very different infectivity, like as you've seen, this virus is much more infectious. Also, the way that it's causing disease is a very complex spectrum of pathogenesis that this COVID-19 disease is causing. So they're not identical, but clearly we can get some really good clues from not only what we have been doing before for SARS or MERS, but also how can we design now a better vaccine or an actual safe and suitable vaccine, and ultimately not only prevent this COVID-19, but hopefully even prevent any future coronavirus outbreaks.
So, now because you have an idea of different coronaviruses, you might move on to producing a universal coronavirus vaccine.
Well, I think ultimately that is what everybody's goal most likely is, to strive to develop something that can be a solution today and fast, of course, but at the same time that it could be a technology that we can also use for many years to come, or even to be the base of an appropriate vaccine that can be then improved or certainly complement additional strategies. I think we all pretty much already have a very key understanding of what are the Achilles heels of this virus and in this family of viruses. As you heard, a lot of people are trying to block the infection by this virus, meaning blocking the entry of the virus into our cells. Mm-hmm. And that's why we're all targeting this so-called spike protein. And even more specifically, in our case, we're even targeting a very specific domain within that spike protein.
So a small fragment of the protein.
A small fragment, yes.
Not entirely, though.
Not entirely the entire virus, because we know that if you block the ability of the virus to bind to the receptor, and the virus uses this receptor binding domain, of course, to bind to the human receptor, we certainly can target the immune response very specifically to block this activity, as well as also reduce any potential of inducing any immune response that is not, one, potentially necessary, or two, that may be inducing some non-desired response.
So what's the progress? How far have you come with developing the vaccine?
So I think we have come very far. I mean, fortunately for us, we have been attempting to develop a vaccine against the SARS receptor binding domain even as far back as 2011. We already have even clinical grade manufactured, suitable for human testing vaccine. It's an RBD SARS vaccine candidate. Considering it is so homologous And we already even have proof that can provide cross-neutralization and cross-protection. We're advancing that vaccine very rapidly. We just forged an alliance with PATH, which is a global organization that clearly supports the advancement of vaccines eventually for global access. They will help us shepherd that vaccine towards the clinical development path. While at the same time, we're also now in our laboratories designing a second generation RBD SARS-2 specific vaccine, but ultimately having both a SARS and a SARS-2 RBD vaccine can even open up our understanding of how we can have eventually a universal or a consensus sequence vaccine.
Fascinating. What about vaccines from other groups?
So we know that as far as we can divide the discussion a little bit in two lines of information. One is, again, what kind of target candidates people are targeting. And you're right. I mean, you can target the entire virus. You can target the entire spike protein, or you can target pieces within the spike protein, such as the receptor binding domain. And therefore, many groups are attempting that strategy. Some groups target the entire virus, some groups target the spike, some groups target the RBD. Then you will ask the question is, when you're saying targeting these candidates, it's how you deliver and what technology you use to deliver such, vaccines. So we know that when you want to target the entire virus, you can do so by using what we call live attenuated or killed whole viral vaccines.
Yeah.
And that pretty much means that you actually take the real virus, you certainly do some modifications in the laboratory to inactivate it or kill it, and that's one avenue. Others take these either spike or pieces of the spike and puts them in what we call types of delivery platforms. Some of them use these viral vector type of platforms, which is using a backbone of a different virus. You kind of modify where you're presenting a viral particle, but using, of course, particles from non-related coronaviruses, and then you insert in your target.
Mm-hmm.
Or in our case, we're using synthetic recombinant proteins that already mimic the natural protein. And then you have these nucleotide type of vaccines, which you're asking your body that, by seeing a DNA fragment or an RNA fragment, you eventually produce the protein of question. So as you can see, we have a wide variety of technologies, a wide variety of candidates, potentially even a wide variety of delivery systems. You can deliver them, of course, either by injecting or by intranasally, by using different formulations of adjuvants.
There is one interesting new technique for injecting vaccines. I think it avoids using syringe, and it uses patches of microneedles.
Correct. So you have, again, very, so as you can see, the ecosystem and it's very, there's a wide array of options.
Yeah.
The one distinction I think that is to make it clear to the community who's going to be listening to this is we can also divide all the attempts that we're doing, between those interventions and those delivery systems and those approaches that we already know have been used before, versus approaches that are also experimental in themselves. And there we look back at how do we already produce vaccines that are already licensed? How do we deliver vaccines that are already licensed? And those are what we consider proven technologies or proven delivery platforms or proven manufacturing process. And unfortunately, only a few of all these different ones that we have already are in that bucket of proven. Recombinant protein vaccines already exist and are licensed. The hepatitis B vaccine, the human papillomavirus vaccine. We certainly already use whole live vaccines, attenuated or killed. However, we do not still have some of these more novel technologies that are already approved and used. And therefore you can already see, right? We have more risk at developing vaccines that we still do not really know whether they work or how you would even make them at large scale versus, of course, maybe those who are already being widely used, and you may already even have an infrastructure set up, not only for its manufacturing, but also of how do you actually deliver. So we need to, so that's, I think, another great value of this great, array of the portfolio, because we are attempting to to direct the fact that, we have to try everything, but keeping in mind the pros and cons of each, the prior experience versus not having a prior experience, the ability of even understanding the cost of goods versus not really knowing the cost of goods.
I mean,
many,
your
right?
group has an advantage of the fact that you have worked with these kinds of viruses before.
Correct. And we have also our approach, because our vaccine center specifically would like to develop vaccines that are for the nonprofit sector, that are affordable, that are scalable, that are, certainly reachable, and that can be made by, many manufacturers. We tend to use, again, these proven technologies already used, that we don't bring any new intellectual property, any new potential question marks of whether they could or not work.
It's like an open source vaccine.
Correct, right. And we know that it can be made. We know that there's a lot of manufacturers that can already adapt and adopt this technology. We have known that these recombinant proteins work, and it's just a matter now of showing that it also can work for a coronavirus vaccine.
Yeah, that is exciting. Well, how optimistic are you about the development of an effective vaccine, and when do you predict it will be ready?
Well, I think we are very optimistic, right? Because even though it's true that developing vaccines is not easy, that it can take years. I mean, some people certainly have this goal of trying to develop them, as fast as possible. We still have to make sure they're safe, immunogenic, and efficacious. We are optimistic because, again, we have so many that, we really need one or two, maybe three, right? And even if one or two or three advance rapidly, all of us behind, or all of us that also join forces, we always can look for how to improve and make vaccines even for the future even better. So optimism is high. The prediction of when is hard to tell. We certainly would like to have something soon. The goal right now, it's yes, as soon as maybe a year or two, but we should not get discouraged if we can do this with a little bit longer time. Ultimately, we need one.
What should people do until then, though?
Well, I think, again, that's where an integrated approach is still essential, right? At all levels, at the level of, having also drugs, at the level of using other immune therapies like monoclonal or convalescent plasma therapies. We should improve our diagnostics. We should improve the health systems. We should improve the access to PPE, right? We should improve the way that we certainly share information, the way that we do social distancing.
Certainly, yeah.
So ultimately, unfortunately, it will have to be, again, one of these approaches that is not going to be that we need to have one single, certainly a magic wand that is going to solve. It's probably going to be the combination of doing a little bit of everything, ideally not, going back to a little bit more social ability of, being together, but at some level, we still need to probably still keep some social distancing throughout this process.
Until it's ready.
Until it's ready.
All right, so since there are like over 120 vaccines that are being developed right now, that means there are many groups around the world that are working on it, some of the greatest minds. What if we put all of them to work in rather limited number of vaccines, so that, by joining forces, focusing resources, we might speed things up rather than scattering energy and efforts, wouldn't you think?
No, well, I mean, there's a value of having so many, right? Because unfortunately, the the ratio of failure in vaccine development at the early stages is pretty high. So I think at the beginning, it's like a big funnel, right? You have to start with a lot of them as they move, because at the beginning you have 80, 70, 80% failure. As you move
Oh,
through the
wow.
process, you may be reducing the rate of failure to maybe 50, then 30, then 20. And then you're right. Then as the big bucket starts narrowing down and maybe saying, these are the ones that have these competencies, these are the pros, these are the cons, then you can narrow the I guess the funnel, and then ultimately you say, yes, these are the top 5 that are going to move forward. I think right now it's too early to just assume which are those top. We have to still bring everyone to the point of where at least they all may have the ability of testing, its ability to show safety and some level of immunogenicity in humans. And then you can see the differences between them. And then you can basically maybe decide which are tier 1, which are tier 2. And unfortunately, as you deprioritize some, you want to have others that raise up, right? So you may want to have even tier 1, tier 2, tier 3s that you can continue moving.
Well, speaking of moving, the virus is also moving, and it appears that it's mutating based on a number of studies. Some say it's 8 strains now, some say it's 30, and some say it's mutations that are key to the virus's aggressiveness, that new strains might be more aggressive. What's your take on this, and how will this affect the development of
Right,
the
and I
vaccine?
think that you're right. I mean, all viruses mutate. I mean, that is certainly not something that we don't know already. I think indeed there are groups that are, of course, specialized in monitoring, the rate of mutation, where the mutations occur, how does it, like you mentioned, have an impact on the pathogenesis of the virus, as how the mutations have an impact on infectivity of the virus. But we vaccinologists should, of course, focus on the mutations that are, again, altering the ability of the binding of the virus to the receptor. Are we seeing any changes there that we should pay attention on? As you know, the virus changes, but cannot really change the things that are essential to it, right? Unless they change it because they want to make the, I guess, the binding to the receptor better, or that gives an advantage to the virus.
Evolutionary advantage.
Exactly. So we're paying very close attention. And I think there is, again, the notion of whatever vaccine we may develop today, we have to keep in mind that we should be intelligent enough to develop something that eventually could be used even in the future for even different sequences of this virus, or even a different member of the family of the same, of the coronavirus. So we should always look for this opportunity of doing a vaccine that could be not necessarily 100% protective for everything, but that clearly confers sufficient a protection, even not perfect protection, but enough to reduce the pathogenesis, enough to reduce the burden, and reduce clearly the severity of the disease and the mortality that this disease is causing.
I see. Well, since once the vaccine is developed, for example, some researchers say that we could speed this up by infecting healthy people. How does this approach work, and would you recommend it?
Well, look, this is not the first time that these so-called control human infection models are used. Many other in fact, we ourselves use one of these concepts for our human hookworm vaccine initiative. But it's not easy, right? It's not easy because the model itself, because it is a model, right, has to also go through optimization and development. So it's not something that you just all of a sudden, implement or incorporate into a vaccine program. Usually it happens when you also do not have a lot of control in the aspects of transmission as far as infectivity. So I think we have to be cautious because of the fact that we also don't have any treatments for this virus yet. Because remember, for example, in our human hookworm model, we know that we can use it because we also have a treatment that can really rapidly be given to the person who gets infected. If you're going to not have something that you can have as a cure, or at least to prevent any seriousness in the actual infection, it would probably not be as ethical, right? We already know the virus is circulating, and we know there are areas where it's circulating at the rates that you can very rapidly see the efficacy of a vaccine. So you always have to question what's the value of adding an artificial, control infection model, and how will you do the actual infection? So again, there's a lot of science that needs to probably be evaluated. I'm not saying that it may not be eventually another aspect of how to integrate within the development process of a vaccine, but it's not as easy as saying, sure, let's just do it.
Let's just do it.
Yeah, we should probably make sure that it is, it has to go through the regulatory processes as a standalone.
Yeah, but in the middle of a pandemic, regulatory processes are sped up. So
true, but they are also already are overwhelmed to try to look at all the data as fast as they possibly can. And regulatory bodies have a mandate to look at these with the eyes of evidence-based, not necessarily to make things faster, but to make decisions, of course, as fast as they possibly can, but based on evidence. And if you don't have enough time to build the evidence, it's a little bit risky to make decisions, right? So you have to balance that, do things fast, but always based on scientific evidence.
That's what should always be the case. Since a vaccine is needed urgently, isn't there a risk of developing a vaccine in such a short notice and trying to get it out there as soon as possible?
Of course, developing biologics always come with that balance of risk benefit, right? And again, it's also be alert that vaccines usually have the highest bar of that risk benefit ratio, because you are giving vaccines most likely to healthy people compared to, for instance, giving a drug or a treatment because you're already giving it to someone who already have the disease or have some so therefore you're looking for something to reduce the burden of the disease versus preventing something, right? So you're right, this is very difficult, and that's what makes the development of a vaccine a little bit longer, and that you have to be a little bit more cautious about it.
So we have to be cautious and probably give it enough time until it's fully ready.
That's correct, and we are certainly doing it as fast as we can. We are, this allows us to look at some innovative ways at how do you interpret data, or how do you even design the studies. A lot of parallel studies, which are of course outside of the business as usual norm. Maybe this would also gives us some lessons learned how to also improve how we just develop vaccines in general, right? But we always have to make sure that safety is certainly evaluated as the ultimate priority.
It's the first step, the most
Correct.
important. Yeah. All right. You mentioned this in a previous question, is the different types of vaccines: RNA, DNA, and protein vaccines. Perhaps you could elaborate on each one of these types.
Yeah, sure. Again, so protein-based vaccines, we already know we've used them for other vaccines. We know more or less how they work. Of course, each vaccine against a specific disease may have a very different mechanism, but at the end of the day, you're injecting the human body with a protein that already resembles something that is very similar to what the natural pathogen has. And therefore you already know how you're kind of like triggering your immune response to respond. With DNA or RNA, you're actually asking your body to do a little bit more work because you want your DNA to then produce an RNA, and then that RNA produce the protein that also ultimately should end up resembling something very similar to the natural protein. So you're just pushing your body a little bit more. You also have to make sure the RNA or the DNA gets into the right cells that have the right machinery to produce these proteins, that then these proteins are presented correctly to the immune response. So again, they may be quite interesting technologies. We know that they have been under experimentation for a while. We know that they've been tried even for many other diseases, HIV, or even some of these cancer type of vaccines. We still don't have an actual licensed vaccine using those technologies. So that means that they may require a little bit more work to establish their real potential. Right?
So the focus would be on protein vaccines.
Well, I think we should focus on all of them, keeping in mind that we have already some precedent of the fact that we know how protein-based vaccines not only work, how they've been working, how they work for other diseases. We know how to even manufacture them. We know how to scale them. We know how much they cost. When all of these, assumptions, we still do not really know them for these newer vaccine technologies.
So if a vaccine that is effective is ready, and many people are not willing to take it, and there are many people, what would be the future of this disease?
Well, you raise a very important point, right? So as we do all these efforts of developing these so-called interventions, we need to make sure that the demand is there, that that they are certainly going to be accepted, that people are going to eventually use them. So that is a very big, important, component to also ensure that they are understood by the population, that they have very high level of safety, and therefore that is the reason why they should use them. So this, we should, this could maybe even help kind of like dismantling some of this hesitancy that people may have, that maybe just be founded on not necessarily evidence. So I think that there's a lot of work that, again, through not only public policy, but certainly dissemination and public engagement,
public health education,
because you're right. I mean, we want to prevent that we lose the faith on the fact that vaccines work. We've seen it. We know they do. We need to reinvigorate the trust on vaccines. And therefore, it would be a big mistake to push a vaccine that we know it doesn't have all these safety,
safety checks
behind, because we can affect also how people may accept other vaccines, which we already see around the world.
I mean, this pandemic definitely showed that a large group of people are resistant to the vaccine, but I think it's our way of getting out of it.
Yeah, so it's an opportunity, but we have to be very cautious of how we take this opportunity in educating and disseminating the correct information, being transparent, make everybody aware when things do not work, so that we can also, they can see how we therefore look for how do we improve so that we can make things work and not only work, but that they keep a high level of safety.
That totally makes sense. And so speaking of safety, what are the risks and complications of becoming one of the first volunteers to be part of a clinical trial that tests a new vaccine?
Well, I think that the good thing is that, again, because it is a very regulated trial, you have access to a very detailed way that they're evaluating you at all levels, right? That again, we have the systems in check. It's not done, in a fast, irrational manner. I mean, there's very specific ways of, how the vaccine is delivered. They monitor, everything of how, you feel, the test that they do. I mean, this is clinical trial science. It's been done for years and years, and of course we're being improving and improving and improving, and we have very, very specific checks. So I think you've seen those who have been very generous at volunteering already, that they're contributing to finding a solution that eventually can then be used for the rest of the world.
That's a
So
very good cause.
yeah, it's to applaud them, of course.
That's right. Um, well, if there is only one thing that we can take from this pandemic, it is that the world was not ready. Yeah. So would it be possible someday in the future to look into the evolution of viruses, predict which one might cause a pandemic in the coming few years, and then start working on a vaccine for that virus, and by the time it causes an outbreak, we already have something ready for it?
That's the ideal, right? And I think we've, in the back of our minds, always had that intent. Unfortunately, a combination of factors, of course, money and just priorities, or even the fact or the realization that we indeed, as a human race, we're not invincible. That, oh yeah, we can deal with it. It's just not going to pan out anymore, right? So
So
again,
it's resources, but visibility wise.
also, we should not forget. I think we human beings always forget very rapidly, right? So we should start looking of how to be less reactive and more proactive. As you said, even if tomorrow we do have a vaccine against COVID-19, let's again continue this effort of, towards a universality, towards what could be other coronaviruses, what could be other flu viruses, what could be other, Zika viruses, what could be other Ebola viruses, right?
Or
And
even viruses that we haven't even studied or learned about.
correct. And not only viruses, right? I mean, again, we have so many other diseases. Again, we in our center dedicate our life to what we call these neglected diseases. They're diseases that have been causing high level of morbidity in billions of people around the world. They may not be emerging, they may not be high mortality, but we have regions around the world that you have billions of people that are suffering, that are not
Yeah.
healthy. And so all vaccines should have the opportunity of being developed, because what you ultimately want is you want a global population with the best possible health health standards, because health then contributes to having better education. Health contributes to having better economic contribution in society. Health also can help alleviate the challenges that our health systems have, right? Because prevention should always trump the treatments, right? Because you want to use treatments only in the event that you really cannot prevent. But if you can try to reduce the need to have to go into hospitals or treatments, certainly that would help. It should be a balance ultimately.
I think you mentioned a very good point about continuing. Um, so, do you think that healthcare systems will change once the pandemic is over to maybe put more funding into scientific research in medicine?
Yeah, we hope, right? I mean, we hope that we see a continuity, we see some sustainability, that we reflect. Maybe, hopefully, not getting into this whole who was at fault or who wasn't at fault, but more than anything is, okay, whatever it is, let's look at how we can improve it. Let's look at how we can make it better. Unfortunately, not every city, community, country may have to use the same approach, but ultimately we all have to look at what are the approaches that work for where we live and who we are, and Maybe that also can help us kind of break a little bit these kind of diversity issues that we have in our world, right? It doesn't
Bring the
matter.
world together to solve such complicated problems.
Exactly, and equitably, and in a manner that it can be reachable to those that have and those that have less.
What are the challenges of producing vaccines in a large scale once it's ready?
Correct. That's again one of the challenges, right? We can have a very nice vaccine. We can, all have, done making attempts at what we call laboratory or pilot scale. Ultimately, you need to have a continuous, constant, large scale production. That's where we have to bring into the game, the big, large manufacturers. There is an ecosystem of vaccine manufacturers globally. Some of them are in high-income countries. Many are in the low, middle-income countries. They have to be ready. They have to be able to produce and have a mechanism of, I guess, selling it at some level, right? Because they're going to need to invest.
This is why your open source vaccine can be a competitor in this case.
Correct, because we don't, our technology will be given to them, with exclusivity, with non-exclusivity, however they want, but we're not going to necessarily want any money for it, right?
So it will be free.
It will be as free as we possibly can make it for free, because we know that this is going to be for, the global public health use.
Yeah. Well, let's hope that it works. I mean, there are many countries that are waiting for it, and
that's correct.
Yeah. Last question. So, any final advice or additional information that you'd like to tell people who are watching this?
Well, I think again is don't be discouraged. We definitely have a lot of groups looking at how to make these, albeit preventive, therapeutic, or diagnostic interventions, or even how to improve the health systems. That said, it has to be an effort that includes even each and every one of us in our communities. Be aware of your surroundings. Don't assume that just because you think you're invincible because you're healthy, and that you're not going to expose others that are maybe less fortunate. That therefore it is going to be a community approach to handle this, and that we hope that we can learn from what all these lessons are bringing to us, and that we can do something better in the future.
Well, let's hope we learn from this pandemic, and let's hope that we will all get our normal lives back soon once the vaccine is ready.
True. Thank you.
Dr. Maria, thank you so much for being with us today and for answering very vital questions about vaccines and the progress with the vaccine. We wish you all the best and with the work on the vaccine, and we hope to hear some very good news soon.
Thank you. I'm too. I appreciate the opportunity, and I hope that we can every day bring new discoveries and new information, and that we will prevail. Thank you.
Thank you so much. Thank you.